Hello my dear students, welcome you all to the biology session. Let us start our topic today. Let us see about the fundamental unit of life. So, the fundamental unit of life is going to indicate the life is about to start from a single cell here. Especially, the cell plays an important role in the formation of complete organism here. If you just see the structure here, you can just see the animal cell where it is going to have all the different kinds of cell organelles and a wall which is going to protect and a nucleus which plays an important role. That the cell is going to have all the different components where ultimately it is going to undergo the processes here. Let us see how the cell is going to be identified and what is the importance of the cell here. While examining a thin slice of cork, Robert Hooke saw that the cork resembled the structure of a honeycomb consisting of many little compartments. So here Robert Hooke, as he is one of the scientists, he has identified the structure of a cell in a particular cork tissue. That is cork in the sense the plant material, or you can just say it as a bark or a piece of plant part which it is going to resemble the structure of a cell here. Next, how he is going to identify the structure is like a honeycomb. You all know that how the honeybees are going to have a U-shaped honeycomb here. Especially in that way, we are going to see formation of a honeycomb-like structure. Suppose if you just see the honeycomb, you can just see a small compartments which are going to be identified here. So, such little compartments are going to be seen in the structure of the cell here. Next, cork is a substance which comes from the bark of a tree. So, just now I told you that it is one of the part of the tree, that is nothing but the bark of the tree. This was in the year 1665 when Hooke made this chance observation through a self-designed microscope. So, how did he identify the structure? As he has said that it is a honeycomb-like structure. So, he has identified it under the microscope. Robert Hooke called these boxes as cells. So, especially each box he has termed it as a cell. Cell is a Latin word for a little room. So, especially here the cell is considered or it is termed as a one of the little room, which it is one of the Latin terminology which Robert Hooke has been identified the structure which resembles to the honeycomb one. This may seem to be a very small and insignificant incident, but it is very important in the history of science. So, the identification of this particular cell structure plays an important role in the entire particular science technology, you can say, because it has made a history in a science that how the cell is going to play a major role. This was the very first time that someone has observed that living things appear to consist of separate units. So, especially here if you consider the living organisms and in that the cell structures, individually you are going to see the unit form. Like each and every organelle or each and every cell part is going to have a different substances where all that undergo the mechanism. The use of the word cell to describe these units is being used till this day in biology. Let us find out about cells. Now here this cell, how it is going to play an important role in the biology, as we are going to see much importance of presence of these cells here. Let us see. What are living organisms made up of? Especially living organisms in the sense, as we are going to see many number of organisms which plays an important role in the survival. So, in that way, actually why we consider these cells as important for all the living organisms, let us see. Let us take a small piece from an onion bulb. So, especially you all know the onion, right? So, how this onion is going to say about the structure of the cell, let us see here. First, take a onion bulb. In that, take a piece of the onion, especially the one. With the help of a pair of forceps, we can peel off the skin called epidermis from the concave side inner layer of the onion. So, especially you can just see the layers of the onion, right? Whenever you cut the onion, you can just see it. So in that you can just see a fine layer which is about to be separated. That layer you need to be collected here. To which side you need to be collected is the concave side. See here, you can just see in the picture that with the help of the forceps how they are about to collect the layer. That plays an important role here in the identification of the cell structure. Okay, that peel of the particular skin part of the onion, we term it as a epidermis. Epi in the sense upper, dermis in the sense the layer which is going to be seen towards the upper side. So altogether we term it as a epidermis. This layer can be put immediately in a watch glass containing water. So after peeling, after removing, we are about to see the layer which is kept in the watch glass, that too especially in the water. Like watch glass is a, like how we have a small plate, in the same way it is made of a glass. In that you need to add the water and put our epidermal layer of the onion cell here, peel, especially onion peel here. Next, this will prevent the peel from getting folded or getting dry. Why do we need a particular watch glass, especially why the peel layer of onion cell is about to be added in the watch glass, especially in the water, is it does not undergo the drying, and you can just observe the cells very clearly here. What do we do with this peel? Let us see. See, if you just observe, you can just see the peel layer under the microscope. As you can just see here, the different boxes, or you can just see here like a brick-like structures which are going to form upon each other. That is nothing but our onion peel layer when it is observed under the microscope here. Let us take a glass slide, put a drop of water on it and transfer a small piece of the peel from the watch glass to the slide. So this is a process actually. So when we add this peel layer, nothing but the onion peel in the watch glass in the water, then remove it from the water and put it on a slide and add a drop of water on it, and you need to be seen under the microscope. Make sure that the peel is perfectly flat on the slide, especially the slide is also one of the glass equipment where it is the one which can be seen under the microscope. So you need to place our peel upon that slide and see that the layer is very uniform, that it is going to fix perfectly on the slide here. Next, a thin camel hair paint brush might be necessary to help transfer the peel. So brush in the sense we are not going to see much bigger one, but a very minute brush which helps in the making of or transferring of our peel onto the slide here. Now we put a drop of saffron in solution on this piece followed by a cover slip. Especially here you need to remember one word, that is saffronin, which we are going to term here as a stain. Stain in the sense a coloring agent which is going to observe the material whatever we are going to place on the slide. As we have taken here the onion peel, right? So towards that onion peel we need to add a drop of the saffronin and put a cover slip. Cover slip in the sense where the peel does not undergo any drying if you put this cover slip. So the layer will be identified in a uniform one, and you can see the layer also very clearly under the microscope. See here, as saffronin is one of the stain which is going to be seen in a red color. Okay, so that you can just see the cells also in the same way. You can just see the border of the cells compared to the previous slide, right? So here this is the cell structure. In this cell structure, you can just see the cell wall at one side, cell membrane in the sense the layer which is present next to the cell wall here, and inside some of the fluid material which we term it as a cytoplasm, and nucleus, and finally the vacuole here, as this vacuole is the one which is going to be seen in the plant cells especially. So these are going to be identified in the structure of the cell. Take care to avoid air bubbles while putting the cover slip with the help of a mounting needle. So all these are like whatever we are going to see, the forceps, brush, as well as here the mounting needle are the equipments which are being used in the process of observation. So here we need to take care while the cover slip is placed onto our slide, because there is a chance of having air bubbles onto our material, whatever we have taken, nothing but our onion peel cell. So this peel layer is going to have a proper way so that it should not contain any air bubbles towards it. Ask your teacher for help. So whenever you are going to do the experiment, you can view it very clearly that how you are going to perform it. We have prepared a temporary mount of onion peel. So this is the procedure that how we have prepared the temporary mount, in the sense for the identification of the structure we are about to be prepared by all these steps. We can observe this slide under low power followed by high powers of a compound microscope. So here we need to see about the microscope structure, as we have different microscopes. So in that, this is the compound microscope which can be viewed the objects very clearly here. So here see, you can just see the presence of the objective lens here, as it is going to have high and low power one, where you can view the objects in different ranges too. So that whenever, see, this is the area where we need to put our slide, especially this is termed as a stage, and beneath you can just see the base. Towards that you do see the presence of mirror. Why mirror is, it allows the light which is being focused from the outside. Either it may be sunlight or it may be the electric bulb one. So it is being focused towards our slide, whatever we have placed on the stage, and it reflects through this lens. And especially this is the view where we are going to see. This is the eyepiece, that where we are going to observe our specimen. Okay, this is the body tube, and these are the adjustments where you need to adjust yourself by your viewing here, so that you can just see the arm. Like for the catching of the microscope, you need a arm here, so that it is being used. And in this way, you can just identify the object very clearly by high power to the low power one. What do we observe as we look through the lens? Can we draw the structures that we are able to see through the microscope on an observation sheet? So now how we are going to identify the structure, let us see. So how you do view under the microscope, the same is the structure we need to be drawn here. Okay, so how do the onion peel cells are going to observe is in the format of this one here. So as these are arranged in a form of a cells which are being present side by side, where inner you can just see the structures very clearly. Individually each cell if you take, see, you can just see the presence of the cell wall, inner nucleus and cytoplasm, as well as a vacuole. All these are the things which plays an important role in the structure of the cell. We can try preparing temporary mounts of peels of onions of different sizes. So by choosing it in a one format, we can go for the preparation of the all different sizes of the onion peel cells, so that you can easily identify how is the structure of the or shape of a particular onion peel cell. What do we observe? Do we see similar structures or different structures? See here in the diagram how you can view the structures. Are they similar or different? So the structure is same, that the presence of the cells which are arranged in a form of brick-like manner.